These are my conflicts of interest. I do have some grant support from the Illinois Society for the Prevention of Blindness, from Google, and the organization Research to Prevent Blindness. And these other conflicts are with drug companies and imaging companies. So I know that many of you are already familiar with what a clinical trial is, and maybe I will be giving you some information that you already know. Hopefully, though, someone will benefit from hearing about this. It's hard to know exactly what information to give you and what you already know. So forgive me if I'm saying things that are already very familiar to you. But what is a clinical trial? It's a study that involves human subjects and a study that's designed to determine if a new treatment is safe and effective. Now, safety is often something that we can learn in the context of clinical trials. But usually at the end of a clinical trial, you don't have proof about safety. Oftentimes you need to wait until later, after a drug is approved, for example, before you can learn more about safety. And I'll talk more about that in a bit. But the main purpose at the end of a series of clinical trials is to determine with some degree of certainty, with a high degree of certainty, that a particular treatment is effective. For example, in lowering the eye pressure, or in neuroprotection, protecting the retinal ganglion cell from injury, even beyond the perspective of eye pressure. So how do you conduct a high-quality clinical trial? One of the key factors is taking subjects and randomizing them into two groups, at least. A treatment group, in which the treatment of interest is being studied, and a control group. And the control can be some existing known treatment that is effective. For example, latanoprost, a drug most of you are probably familiar with, that we know lowers eye pressure. If we want to compare a new drug to latanoprost, a great way to do it would be to randomly assign patients to the new drug or to latanoprost. And then the other critical element is masking. So you've heard of double-blind clinical trials. We don't like that phraseology in ophthalmology. So we talk about, we don't like that at all. We talk about double-masked clinical trials or single-masked clinical trials. So in a single-masked clinical trial, the investigator who's studying the particular treatment is typically masked or unaware of what treatment assignment each individual patient being studied has been given. In a double-masked trial, both parties, the investigators and the subject, the study subject or the patient themselves, are masked. So for example, if you've ever been in a clinical trial looking at a drug study, looking at a particular drug, you might find that the bottles are simply labeled A and B, and you don't know whether you're getting the study drug or a comparator or maybe even a placebo. Usually, when you start a clinical trial, I shouldn't say usually, you'll always be told whether the comparator is placebo or an active drug. We'll talk more about the ethics of conducting a clinical trial in a moment. So some other different types of clinical trials, some other characteristics that may vary. For example, you can have a single center or a multi-center clinical trial. We at Northwestern could decide to do a clinical trial looking at the effectiveness of something like resveratrol to see if it's neuroprotective. And we could conduct that in our own institution. Or it could be a multi-center clinical trial in which we team up with 9 different centers throughout the country to try to study the same question, because it's hard to do it in just one place oftentimes, depending on what the question you're trying to answer is. And many clinical trials are investigator-initiated. For example, if I want to know, it's my idea, and I want to know whether resveratrol works, then that's an investigator-initiated clinical trial. We can seek funding, but it's not sponsored by the federal government or it's not industry-sponsored. So that's the other major and most commonly conducted clinical trials, actually industry-sponsored clinical trials. A drug company has developed a new drug and they want to prove that it's as effective as timolol, for example. That could be a clinical trial. And then there are these very large, federally funded, very important multi-center clinical trials like, for example, the ocular hypertension treatment study, which I'll talk about in more detail, that many of you have probably already heard about. These are really important clinical trials that have taught us very important information about glaucoma and how best to treat people. And really, these are the kinds of trials that have proven that lowering the eye pressure actually works to help patients with glaucoma. Now, you might be curious about phase 1, phase 2, phase 3 clinical trials. So these are FDA clinical trials. So when a company wants to get a drug approved for clinical use, they start with a phase 1 clinical trial. These are small studies. Typically, you have about 20 patients, and you're trying to get a general idea about safety. And you want to get a hint about effectiveness, but the key thing is trying to find out whether it's safe to unleash this new investigative drug into a larger cohort of patients. You also want to know about dosing, and you can answer that in a phase 1 clinical trial. In a phase 2 clinical trial, you want to get more information about safety and more information about effectiveness, and you're studying more people, and you know that you can do it because the phase 1 clinical trial showed you that it was safe. And then the phase 3 clinical trial is the gold standard that the FDA uses to determine whether a drug should be approved by the FDA for use in the United States. And here, the FDA and the drug company collaborate to come up with a plan as to how the study should be done and what endpoint should be used and how many people should be studied. And a successful phase 3 clinical trial will typically culminate in the approval of a new drug or a new surgical device. A phase 4 clinical trial, that's after a drug has been approved. You still want to keep an eye on things. You want to do some additional testing to see if it's better in certain types of glaucoma, for example, or you want to do some additional testing in children. Those are phase 4 clinical trials. So the nuts and bolts of it, how does it work? So first, you've got to design the clinical trial. And typically, what you have is a carefully designed set of inclusion and exclusion criteria. You want certain patients who have certain characteristics. You want to make sure you don't enroll anybody who may be harmed by enrollment because, for example, their glaucoma is very advanced and you don't know if the new drug works yet. So you don't want to enroll them in a trial where they're going to be stuck, possibly with a pressure that's too high. So an example, if a patient has high pressure but no glaucoma yet, you know that, and the pressure is 24 or higher, no higher than 30, then you know that that patient, this happened 20 years ago, may be somebody that you could just monitor without treatment. We don't know yet. Is it beneficial to treat patients like this? Or we didn't know in the late 1990s when this important clinical trial was started. It's called the Ocular Hypertension Treatment Study. So patients were randomly assigned to either no treatment or treatment, and then they were monitored very closely every 4 months to look to see if their visual fields became abnormal, to see if their optic nerve changed shape in a way that suggested that they had glaucoma. And then, at the end of all this, the outcomes in those 2 groups, there were 800 people in each group in that study, to see if the outcomes differed. And then you publish the findings to disseminate the information so everybody can use this information to better take care of patients. And along the way, you know, there's also, in the beginning, review by a human research ethics board. So at Northwestern, we have our own IRB, and they are comprised of experts and members from the community, lay members from the community, non-scientists, who look at a clinical trial and ask the question, is this an ethical clinical trial to conduct? Is it okay not to treat people who have high eye pressure? And then, while a clinical trial is ongoing, there's a special committee of experts, a data safety monitoring committee, that looks at the data as the trial is going on to make sure that no harm is happening to one of the groups. And then, of course, like I said, statisticians are involved in analyzing the data, and then the results are published. So why do we need clinical trials? If it's not already obvious, I'm going to give you a concrete example that I think is really interesting. So this is a surgical device called the CyPass. So the CyPass was designed to be implanted into the front of the eye where the fluid exists in a space called the anterior chamber, and it allowed the fluid to gain access to this space from where we know there's a huge reservoir, a huge potential space for the fluid, and that the fluid can be easily absorbed from there. We know from old surgeries that opening up this space can bring the pressure very low. So this was a great idea, and in fact the inventor is a friend of mine, Eugene Dewan, and he drew this for me on a cocktail napkin back in 1999, and then went on to build the company and sell the company that developed this device. So this underwent clinical trials, and this was approved by the FDA and implanted in many people. And what was observed in the clinical trial was that the stent was effective. This was a 2-year clinical trial, and it showed that compared to baseline, it lowered the pressure an average of 7 to 8 millimeters of mercury at 12 and 24 months. Sounds great, doesn't it? But what if the clinical trial had been done just like this? There were no control. We would think that there was this extraordinary pressure-lowering effectiveness here. Now, what I didn't tell you is that when this clinical trial was done, the way it was done was patients underwent cataract surgery at the same time that they underwent implantation of the CyPass device. So here is the control arm. These are the people who just had cataract surgery. So they were randomly assigned to just have cataract surgery and not have implantation of the new device. So that's the blue group, and the red group is people who had both cataract surgery and implantation of the device. And now we learn that the actual effectiveness of the device is only about 2 millimeters of mercury of pressure lowering. So that's a really important example, I think. Now, the FDA said after they approved this device, okay, it's approved, but you've got to keep track of these people and make sure it's safe, even though it's approved and even though at this point any ophthalmologist can implant the device. So what was discovered about 5 years after the clinical trial was completed was in the blue group, which in this example is equal to the red group in the previous slide. These are the people who had both the CyPass device and cataract surgery. There was more death of corneal cells that keep the cornea, the front clear part of the eye, clear. And the FDA declared this class 1 recall situation. So this device was recalled, not when already implanted, but from the shelves, and this device could no longer be implanted in the United States. And the company that owned the product appropriately removed the product from use too. They did that voluntarily before the FDA required it. So this is a great example where clinical trials can provide you really important information that helps us take better care of patients and that helps keep patients safer. So, how are study participants protected? So all clinical trials that are conducted in Western countries must adhere to certain rules. And in the United States, on top of that, there is protection afforded by HIPAA, the Privacy Protection Act that was passed in 1996. So the Declaration of Helsinki declares that humans have the right to know what's being done to them in these kinds of clinical research settings, and they have a right to agency and to make determinations about what will happen to them, right? And the US law requires that there is an ethics oversight committee that monitors all research that involves human subjects. And again, like I said, privacy is managed by and regulated by HIPAA. So I think that in the current climate right now, and this may not have been as well the case, let's say about 20 years ago, that we're all very, very careful about this, that regulatory bodies have very strong authority to control what happens and to investigate what happens and to make sure that everything is being done correctly to keep patients safe and informed about the risks of clinical trials. So I'm going to go through, Paul, if I may, can I take a few extra minutes here to talk about some clinical trials, some specific?
Okay.
positive information here. So I gave you one bit of bad news that was learned in a clinical trial. So let me give you some good news. So this clinical trial was done in the UK. This was a great clinical trial because in just 2 years, the investigators were able to figure out that latanoprost protects the visual field in patients who have glaucoma. This is the strongest proof that we have that lowering the eye pressure helps. So that might seem mundane to you, but it's really important because this is a placebo-controlled clinical trial that showed that in a large cohort of about 1,000 patients, that lowering the intraocular pressure with latanoprost specifically protects the visual field. I'm sorry, it was 500 subjects. Okay, so here's another clinic, and that was a federally funded in the UK clinical trial. The ocular hypertension treatment study, which I mentioned, I think this is probably the most important clinical trial in terms of what we have learned. So this was the one where people with high pressure who did not have glaucoma were randomly assigned to either treatment or just observation without any treatment. And what we learned is treatment definitely reduces the risk that these patients with high pressure will go on to develop glaucoma. But we also learned that 5 years after diagnosis, only about 10% of patients who had high pressure actually go on to develop glaucoma. And we also learned that delaying treatment did not cause a penalty and did not make people worse off. So maybe in some situations, it's okay to monitor patients with high eye pressure and initiate treatment later. And even more importantly, and that was because at the end of the 5 years, patients continue to be monitored. And we just got 20-year results from that clinical trial recently. And I think most importantly was this. We learned, and I've written on the right side of the slide here, 5 important things that were the driving risk factors at the beginning when you enrolled the patients that could be used to predict the future risk. So now we can take patients who have high eye pressure, and we know that the higher the eye pressure, the higher the risk that they'll go on to develop glaucoma. That's pretty intuitive. The thinner the cornea, the higher the risk. And that used to not be intuitive. And that was a new finding that was really revolutionary. A lot of us suspected it even in the late 1990s, but this proved it. The older the age at the beginning, the higher the likelihood the patient would go on to develop glaucoma. The more damaged looking the visual field, even though it was normal, the more damaged looking the visual field, the higher the risk that the person would go on to develop glaucoma. And the bigger the cup in the optic nerve, I think most of you probably know about the cup of the optic nerve. The bigger the cup, the more likely the person would be to go on to develop glaucoma. And the cup is a part of the optic nerve where in glaucoma it gets larger. So now what we can do is we can take a patient who has high eye pressure but they don't have glaucoma. We can plug these numbers into a computer and we can predict the 5-year risk that the person will go on to develop glaucoma, and this can guide us as to whether or not we should treat that person or just keep an eye on them. And I think I'll skip this one because I've gone quite a bit, but we've learned a lot from surgical clinical trials too. These are some new results that I was excited about when I put this talk together, but I don't think I want to take all that time. I do want to talk about how you can help. So selective laser trabeculoplasty, SLT. So in the UK, a study was done that compared for newly diagnosed patients with glaucoma and high eye pressure, what's the best treatment? It compared medicines versus laser, SLT, selective laser trabeculoplasty. And the 3-year results found there was really no difference. The groups did pretty much equally well. There was a slight advantage for the laser group, but it wasn't dramatic. Well, I've seen some additional results, and the newer results, the 6-year results, conclusively prove that laser is superior. So laser treatment for glaucoma, initially diagnosed, is better than medical treatment. So we're doing a clinical trial at Northwestern. We're part of a group of a federally funded clinical trial called the COAST study, in which we're evaluating SLT in more detail, trying to see what the best dose of the laser energy is, and trying to see whether annual low-dose treatment is better than as needed regular dose treatment. So how can you help? Many of you who are in this room, you may already have been treated. If you've already been treated, you can't be in this clinical trial. These are newly diagnosed patients, but I'm sure that your friends and family members, anybody who's been newly diagnosed with glaucoma may come to you and ask you questions as to what they should do or to learn more about the disease. And you can tell them about this clinical trial, COAST. So this is my email address. If anybody has any questions, I'm happy to address them.